All right, so we're going to go through transcription and translation, focusing on new elements from bio or honors bio, and elaborating on those. So some things I'm going to go over fairly quickly, good review. Please note that along with this video, the actual PowerPoint will be uploaded on Google Classroom. There are a lot of links to videos in the PowerPoint. I'm not going to play those throughout the presentation, but those are definitely something you should go back and access. There is a couple recap questions at the end. We're going to go over those at the start of class next time. And there is also a practice quiz from some previous work that will be posted on Mastering Biology, along with a quiz for this material posted next week. So keep your eyes peeled for that. Okay, so what do you need to know? We're going to focus on things like gene expression. So how do we go from DNA to actual expression, and then connect that to phenotype later. Transcription and translation. We're going to talk a little bit about DNA versus RNA, the events of transcription. We're going to compare and contrast eukaryotic cells to prokaryotic cells. We're going to move on to translation, and then we will get to mutations in class a little bit later. I'm not going to talk about those in this video, but we will talk about those in class later on, and they are on the PowerPoint, which you could access on Classroom. Okay, so big concept in biology is the central dogma, which talks about this three-step process of taking information from DNA, converting it into an RNA format, and then using that to create a protein. So that is really what we're focusing on here, breaking that down to transcription, which goes from DNA to RNA, and then translation, which goes from RNA to the protein itself. Knowing that during translation, this is going to happen on a ribosome. A lot of experiments to test this over the years and figure out that there's one gene that goes for one protein within in a eukaryote or prokaryote. So this Beetle-Tatum experiment, pretty classic. You saw this on your DNA timeline. So they were using X-rays to trigger mutations, and they noticed that when they placed the mutated DNA into a nutrient broth, so providing it with the components that it needs in order to make proteins, they noticed that only the protein affected, which tied back to the area of DNA that was mutated, was unable to be produced. So they really were able to connect that there's one gene for one protein. Now, do keep in mind that when you're talking about proteins that make it all the way to quaternary structure and have multiple segments that connect together, those are playing off more than one gene. So there are some complications to this, some more, as in all science, it depends. So in cases like that, for something like hemoglobin, there could be a mutation that affects one segment of the protein that comes together to make hemoglobin, but maybe not the others. So there are some exceptions to this. All right. Bacteria prokaryotes versus eukaryotes. So you can see these are overall diagrams of transcription and translation. I'm going to point out a couple things, but as we go through in more detail, we'll point out the real variances that occur here. So obviously we know structurally the differences. In our prokaryote, we've got our DNA, not in a nucleus, so it's exposed to the elements of the rest of the cell. So we're going to go ahead and go through transcription, and we're going to read the DNA to make RNA. That RNA is directly going to go out to the ribosome to instruct it to make a protein. However, in eukaryotes, as in cellular structure is more complicated, so is this process. There's a lot more going on. It's not that simple. It's just transcription, translation, that's it. So in bio, that's all you really focused on. So we're really going to add in all the complications and elements of control that are found within eukaryotes that we don't find in prokaryotes. Some of those include modification of that RNA. So the RNA that's created in transcription is not the same one read by the ribosome to make the protein. There's modifications made to that, and then also steps that help get it to the right ribosome for protein creation. Okay, structurally, Imagine you remember most of these. So DNA, we've got nucleotides just as we do in RNA. However, DNA is double-stranded, RNA is single-stranded. Big difference here structurally. In DNA, we have the deoxyribose as our sugar, where RNA has ribose. We know that only thymine is found in DNA, where it's not found in RNA. However, the other three are universal between the two. Thymine is replaced by uracil, and RNA is really that secondary messenger instead of the original template. There are a lot of different types of RNA. I'm not going to sit here and read these to you, but they're going to come up. I just want you to notice the diversity of jobs that RNA has in this process. So it is really critical and used in a variety of ways. There's theories out there that RNA was the original genetic material and that DNA came along later. And that's something we'll talk about, what are the benefits of DNA versus RNA in these different roles, but it really is a large component in all of these processes, especially in eukaryotes where things are much more complicated. All right, so just as in DNA replication, we've got one side of the DNA that is a template strand that's going to be read. Same rules, it's read in the 3 to 5 prime direction. That we're going to have here the complementary strand built off of that in RNA form. So anywhere there would have been a T, a U will be in place. We go off to translation. That will be read in codons or chunks of 3 that are going to code for an amino acid. Okay, here's our amino acid chart. We know there's 20 different amino acids that we have. By reading in groups of 3 like this with a potential 4 different letters that could be in each place, we end up with 64 possible combinations. So there's a lot of redundancy that does help to reduce mutation risk. A lot of that redundancy shows up here in the third placeholder in the codon. So if you'll notice here for serine, it doesn't really matter what the last letter is. If it starts with UC, it's going to code for serine. So we'll talk a little bit later when we look at translation about wobble, and that's going to help reduce mutation risk over time. All right, so let's dive into transcription a little bit more. This I've already mentioned here at the top. Big thing I want to point out here is at the bottom was that we have some additional elements that we kind of ignored in bio, things like the promoter. So here I'm going to look at bacterial, and then we're going to switch over to eukaryotic when it starts initially doing transcription. So we don't just start at a point and just go. We actually have a promoter that binds to the DNA. You can see it here in this diagram, just ahead of the starting point in terms of what needs to be read and copied into RNA form. Now, in bacteria, that's pretty straightforward. It's just a promoter, it forms. However, in eukaryotes, it's a little bit more complicated. So in eukaryotes, the promoter region's a little bit larger, and there's more factors that bind to that area before transcription can actually take place. So something that we look for in eukaryotes is called a TATA box. That TATA box is upstream or found above where the promoter is going to bind. And transcription factors, which you can see here in the second diagram, will bind to that TATA box and form a complex. And then that is going to essentially signal or flag where the promoter needs to bind. So you can see here in this last picture, the promoter binds over where these transcription factors have bound to different coded regions along the DNA upstream of where transcription has to occur. So something that's different in eukaryotes, a little bit more complicated, there's more molecules involved, and something we hadn't talked about in previous classes. Okay, what isn't really different is the actual transcription with RNA polymerase, following the same basic procedure and rules that DNA polymerase follows in DNA replication, obviously with the exception of using uracil instead of thymine. So you can see that occurring here on the left. So that promoter will allow for RNA polymerase binding, and it will travel down the strand until it reaches a sequence that signals ending of that transcription process. Okay, so the terminator is shown here, not to be confused with Arnold Schwarzenegger. So it's going to go ahead and it's going to travel down. It's going to hit that terminator sequence. Again, a chunk of code in the DNA that's going to signal the end of this process. It's going to essentially fall apart. That whole complex will break down and the RNA will be released. Now, in eukaryotes, we don't actually call this messenger RNA at this point. So in prokaryotes, it would be messenger RNA. It would go off to translation. But in eukaryotes, it's actually pre-messenger RNA because it has to be modified before it can be used. Okay, so what are some of the modifications that occur? So in this pre-messenger RNA, a couple of things are going to happen. We're going to add on a 5' cap. That 5' cap is going to be up above of really the coding region, what's going to be read by ribosome in translation. That 5' cap is going to do a couple things. It's going to help export it from the nucleus, get it where it needs to be, but it's also going to provide protection. There's a lot of harmful negative elements out in the cell that could potentially affect or damage this messenger RNA, and we don't want the code that we need to read to be damaged or altered in any way, or it won't result in a functional protein. We also have a poly A tail. Again, a lot of that is for protection. And helping it get to the correct ribosome. So what we really care about is the protein coding segment, the chunk of RNA that is actually going to code for the protein that we want. However, there are modifications that are still going to go on there as well. So not everything that was copied during transcription is necessary to make the protein we want to make. So there are areas of this messenger RNA that is labeled as introns and exons. So exons have to exit from the nucleus. We want those pieces. They're in darker red here. The introns are not needed for the protein we have to make in this instance. So they're actually going to be cut out, and those exons will be bound together to make a shorter coding segment. Now you might be like, well, why? Why would they do that? It doesn't make any sense. Consider the ability to have a code for multiple proteins in the same region, just requiring you to cut in different places. So you actually could have codes for proteins needed in similar processes within the same chunk of DNA, just depending on how you slice the messenger RNA. So keeping those codes all together can help protect and insulate them, making sure you only have to initiate the transcription of one area and then just cut in multiple ways. So splicing and spliceosome will take care of this work and will be shown here on the next slide. So it's going to essentially have a signal here and towards the end to know where to start and to stop. And it's going to allow it to bring those pieces of intron together and then cut and like ligase, put them back together or bind them together to seal the gap once they've sliced out the pieces that aren't needed. Okay, we talked a little bit about the benefit of that. So we're going to move on to translation. Translation, something you've covered. We're not going into much more detail with that. We know that we have transfer RNA shape here in kind of this T shape. And there's two critical ends to that transfer RNA. It is the anti-codon end, which is complementary to the codon in the messenger RNA. And then there's the amino acid attachment site. So the piece that's going to hold the amino acid that this anti-codon codes for. So the two critical components. Now, mentioned that wobble earlier. So quite often, if you look at the amino acid chart, that third letter is not as critical. So what can happen is sometimes really these first two letters are critical, and then there's some more play, if you will, in the binding of this last one. Where as long as it starts with AA, quite often we can bind correctly to the codon, even if this last letter is technically improper. So again, that redundancy you'll see a lot with the first two letters coding quite often, regardless of the third letter for the same amino acid. Okay, so when it comes to the ribosome, there's three critical areas on the ribosome where this process is going to occur. So this is a more molecular view, but we're going to switch over A different view here. It's more animated and easier to follow. So ribosome has two pieces, the small subunit and the large subunit. On that larger piece are three binding sites. So when we get started here, at the A site is really the attachment place. So that's where the first transfer RNA is going to bind to the messenger RNA, is going to attach in the first place. Okay. The P site is kind of the holding site. So the polypeptide, the protein that's being made, will always be held at the P location. And E is exit. So once you're done, you slide over and you break your bond and you're off to go find a new amino acid. So you can kind of see this below. We've got this growing protein here at the P site being held by the transfer RNA. This new transfer RNA comes in with its amino acid. They're going to bind together. This middle one's going to release its growing protein, let it be held by this one currently in the A site, and then everything's going to shift over. So this first one will be in the P site. This now empty transfer RNA is going to switch to the E site and be kicked out to go get a new amino acid. All right, so you can kind of see this in sequence, what I just described here. You'll notice what is required for this. We're making bonds, so energy is required. So you can see here, it's not actually ATP, but GTP, which functions in the same manner, coming in to provide the energy to form the bond in the protein. All right, eventually we're going to reach a stop code. So we talked quite a bit in bio about there's nothing actually coded for in that stop code. So that stop code causes the binding of a release factor. So the entire complex essentially falls apart. The polypeptide, the protein, is released to go off and fold. Transfer RNAs are all released to go get new amino acids, and the small and large subunits of the ribosome will break up along with the messenger RNA. Now, if we need a lot of a protein, it's not necessarily a one and done type of thing. Many ribosomes can come along and read the same messenger RNA to build multiple proteins sequentially. So if you need a lot of a particular signaling molecule, many of those could be built off the same messenger RNA. Messenger RNA actually has kind of a built-in destruction code so that it does break down over time when it's not needed anymore. We've talked about this a little bit before, but we know we have two different areas for ribosomes. We have free ribosomes and bound, the ER. So those free ribosomes are going to make proteins that are going to stay within the cell, where the bound, because they're a part of the endoplasmic reticulum, which leads to our endomembrane system, those generally are going to end up on the cell membrane itself or released from the cell. Okay, so in that initial modification of messenger RNA, we did have a signaling molecule that helped it get to that correct ribosome that allows it to know whether it needs to be on a free or an attached ribosome. And here's just a modeling of the work going on here in the cytosol versus the endoplasmic reticulum to go in for that protein to be folded and modified. All right, so here are some recap questions. So you should pause the video here. What I challenge you to do is go ahead and answer these questions. Be prepared to discuss these in class next time. It'll kick off our discussion. I'll answer your questions. We're going to do an activity or two to make sure that you've been able to kind of synthesize and understand and apply this in a couple different ways. And then we will move on and just kind of determine our best path from there.